Don't know me, and I am the co-chair of the biotech program here at SEC. And today we are going to have Dr. Nicole Buan present to us, and she comes to us from the Department of Biochemistry. Is going to tell us a little bit about her research with biofuels. So I'm super excited to hear about that. And Nicole,
Great. Thank you, Tracy. Absolutely. Okay, so today I'll be telling you about fueling the future and how we engineer our organisms of interest called methanoarchaea. So I'm what I would call a microbial physiologist. Um, and that's like really kind of a really kind of niche specialty that's really interdisciplinary. Uh, Microbial physiologists really think about what do cells need to grow? How do they grow? In other words, how does metabolism occur? What's the mechanism of that? And then we often use synthetic biology really to ask the question, how can we make cells grow better? Okay, so there's a lot of applications of this, of course, in the microbial world, but you can imagine you can apply this to thinking about how microbes interact with plants to get plants to grow better, so on and so forth. So this is really a transdisciplinary field, and we're really trying to identify even general principles that apply to our organisms of interest, but maybe even more broadly than that. So if we think about biology, it's actually really complicated. And we have to remember that cells are not trying to make us happy, right? So if you're a microbial physiologist or if you're trying to engineer microbes, they don't do what we want them to do necessarily. What they're really concerned about is what they need to do to be able to grow and divide, right? And in biology, or as we think about science, we often reduce the complexity of biology to very simplistic terms because it is so complicated. We're like, okay, just give me this one parameter to look at, right? So we tend to ask and we tend to design our experiments to tell us what is necessary and sufficient, like what's the minimal to get a particular phenotype or to get our cells to grow. And we try to almost like eliminate redundancy. Okay, if there's two genes that do a similar thing, we kind of try to ignore that, or we tend not to think about that. But redundancy is actually really important in nature, right? You can have overflow pathways or complementary pathways. And so we, as a microbial physiologist, we need to understand when is something necessary and sufficient versus when is it redundant, and why does the cell care? So we have to remember that the goal of biology is not necessarily to be just the most efficient or the most perfect in our eyes, the way we want to think about it, but really merely to exist. Okay. Another thing that I want to mention is biology is really, really complicated. And even when we think we're working on like one type of cell, like in a pure culture, you have to remember that there's billions and billions of individual microbes in that culture, right? And so even though they might be genetically very, 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 very similar, there's going to be point mutations. Or depending on where those cells are in the culture, or if they're in a biofilm, in a skin type of thing, they're going to be doing slightly different things, even though they have very similar or identical genetics. So there's many ways to get from A to B. This is the beauty of biological diversity, even when the genetics seems almost identical. And so what cells are really caring about is, let's say that they are growing on glucose and then producing ethanol. So they're turning glucose into ethanol, and that's the way that they're surviving, right? They're going from A to B in terms of metabolism. In biology, there's a lot of ways to get from A's to B, right? So that's the beauty of biological diversity. There are many, many different ways. Some of those are more efficient than others. Some are highly efficient, some are pretty wasteful and kind of sloppy. The cells don't care as long as they can grow and produce progeny and survive to the next generation, right? So as a microbial physiologist, we have to keep all of this in mind, even as we're learning about the genes, the enzymes, metabolism that's going on. So biology has mind-boggling metabolic complexity. This is really famous picture of E. coli's metabolism, all the different metabolic modules, fatty acid biosynthesis. This is the TCA cycle that we learn about in biochemistry a lot. We talk about it. There's thousands of reactions, 5,000 genes, even in the very simple E. coli microbe. It's almost too mind-boggling to think about all at once. And in fact, this is why people are feverishly trying to use AI to try to figure out even simple cells. Okay, so there's a lot. But amongst all of that complexity, a microbial physiologist looks at that and goes, but you know what? There are some physical and chemical constraints on all of that, all of those possibilities that really define or constrain the behaviors that we see that we can observe. And as a microbial physiologist or a metabolic engineer or a synthetic biologist, our job is to manipulate all of these different factors, these different environment, like the temperature, water activity, pH, or the information, which are the genes or how sensors in the cells work, as well as the components in the environment, so trace metals, the amount of carbon, nitrogen, phosphorus, all of these ingredients. We manipulate these, and then we see how the cell responds, how fast do they go, and how organized the cells are. So this is what a microbial physiologist kind of thinks about when we're thinking about metabolism. And if you say, I want an organism that's going to be better than yeast at making ethanol, these are the kinds of things that we're thinking about. Are there other genes that we could use or that some other organism might have? Maybe they're using a different sugar, or maybe we want them to grow on a completely different carbon source for our industrial process. Or maybe a better way to get them to produce ethanol is to control these environment vectors, the amount of oxygen that's available, things like that. So the organism that we focus on the most in my lab are called methanoarchaea. So everybody knows, about E. coli. There's a lot that's been done for 50 years. They don't know that much about these methanoarchaea, but there's actually a longer history of these organisms in the scientific literature, but they're really difficult to work with. So these organisms grow by making methane gas. And that is crazy because they're using these low energy substrates like hydrogen and CO2, acetate, which is just a 2-carbon compound, or formate, which is a single carbon compound. And they're using this pathway, these enzymes, which we call the methanogenesis pathway. And while they're growing, they're taking these low energy substrates and producing a high energy product that kind of seems to totally break the rules, actually. So methane is what they're producing. And this is a high energy fuel that we can use for lots of purposes. So it's the same thing as natural gas. It's the same thing as biogas, okay? So these methanogens can grow on these low energy substrates. And then actually, through our studies of environmental microbiology, we're finding more and more signatures of these methanoarchaea in the environment. And we think also that they can grow on potentially things like coal or other compounds. We know they can also grow on carbon monoxide gas, okay? So they can grow on these low energy substrates and produce a high energy product, methane. And people don't necessarily hear about it that much, but it's a significant percentage of the global carbon cycle. About 2 gigatons of methane are produced worldwide all the time, every year. So if we could harness these organisms and the methane that they produce, we can sort of almost like, for our purposes, accelerate the global carbon cycle, but produce a lot more renewable natural gas instead of having to rely on fossil natural gas. So this is what some of these organisms look like. They're actually evolutionarily quite diverse. So here I'm showing my favorite methanogen. That's also what we call them, methanogens. Methanosarcina acetoborans, which are these cute little spheres. In some ways, they're not that interesting because they're just a sphere. But there are other methanoarchaea in the environment that make spirilla, or more like rods, like all of the different things you learn about in microbiology class, all those different types of morphologies. So a lot of them are the one that we prefer to work with, form these nice spheres. We can grow them as single cells in the laboratory, but they might also be found in clusters in the environment. So these methanogenic archaea are the dominant archaea. So they're not bacteria, they're archaea, and they dominate in low sulfate sediments. An example of that might be a freshwater stream like you see here. I took this picture in Rocky Mountain National Park in Colorado. So where it is anaerobic, these microbes might be found in that subsurface. So they're consuming the acetate or formate that are produced through fermentation from all of that biomass by all the other microbes. And then they're consuming that and converting it into methane. And there's a lot of reasons to study methanoarchaea. They are part of the global carbon cycle, which I mentioned. They produce this renewable natural gas. We're pretty excited about that and optimizing that process for industrial needs. But they are also the part of the flora of higher organisms, including ourselves. So when you hear about, so we have these organisms in our gut microbiomes. It's a small percentage, but they're persistent and they have actually evolved with humans. So we know that. We have specific human gut methanogens that are in there all the time. And of course, you probably have heard of methane being produced by cows, right? They burp it up from their rumen and they have methanogens that have evolved along with them. And it turns out people are trying to uncouple methanoarchaea growth or methanogenesis from rumen fermentation. And it turns out when people have tried in the past to inhibit these methanoarchaea, it actually negatively affects the cow. So they're helping the cow to actually metabolize their feed more efficiently. Okay, so lots of great reasons to study these organisms. And I mentioned that 2 gigatons worldwide are produced. This is a picture in Canada. So in that subsurface aquatic, low sulfate type of environments, you can just get an idea of just how much methane and CO2 can be produced by fermentation. A lot of these bubbles that are captured in this ice are actually methane bubbles. Okay, so it's actually a significant amount of methane flux that ultimately can make it up into the atmosphere. The majority of that methane that's produced in the subsurface is actually consumed by other microbes. We call those methanotrophic microbes. So they eat the methane. So that's good because methane is also a potent greenhouse gas. So in nature, it's normally quite balanced. Humans would like to sort of unbalance it to either prevent as much production in the environment. That might be hard though, because you're talking about like the earth, they're everywhere, including in us. But in a tank, right, where we could maybe capture and do anaerobic digestion, if we could enhance methanogenesis in a tank, we could convert all of that biomass into methane a lot faster and use it as a fuel. So that would be a good thing for us. So my lab does methanogen engineering. We use, these organisms are inhibited by exposure to oxygen. So we can do just about everything you can do with E. coli, but we have to do it in these anaerobic incubators and gas chambers. So here are vinyl chambers, which we love, and we have to put our hands through the gloves here. And inside here has only trace or undetectable amounts of oxygen. So we actually have chemical systems that scrub off any oxygen. So it's not even enough just to put it in plastic. Turns out oxygen gets through plastic, maybe a little slower, but it gets through. So we actually have to have catalysts in here to even continuously scrub out the oxygen that's in our chambers. And we grow them on anaerobic culture media. So we have to sparge in nitrogen and CO2 gases to displace any oxygen that might have dissolved in the culture media. And we put a color indicator to tell us not only that the oxygen is gone, so here you can kind of see that, the oxygen is gone, and then we also add a chemical reductant, in this case, cysteine and or sulfide, so hydrogen sulfide, and that removes any last trace of oxygen. And then we can dispense that into anaerobic vials, bottles, and these long skinny ones are called bulge tubes. And that's how we grow the microbes. In this chamber system, we also have wolf anaerobic chambers here. So there's a chamber within the chamber. And that's where we can grow them on agar plates, very similar to how you would with E. coli. So we can do everything, and we can transform cells to add plasmids and so on and so forth. And that's the kind of thing that we do in our lab. So I'm going to tell you a couple of stories about the science that we do. So I really try to think about, again, as a microbial physiologist, I'm like, how can we make them more efficient? What if we like poke it here or tweak it there? Can we make it less efficient, or can we turn that carbon more into biomass, but not as much methane, or vice versa, or make super methanogens? Like, we just want to tinker with it and see what we can do. So this particular project was carried out by a postdoc a few years ago, Annalise Duclouseau, and an undergrad, Nicholas Dushenko. And we were asking the question, can we minimize kind of like kinetic entropy, which is like the speed of the reaction. So even in these really amazing cells that can, produce methane, and it seems to break all the rules, they're evolutionarily ancient. So we think they're really, really efficient. But even then, nothing in biology is perfectly perfect all the time, right? So are there ways that we can tweak it? Can we make it go faster? So that's what this project was about. So this is what the methanogenesis pathway looks like. So you don't have to learn all of this. It's pretty complicated. But if you remember when I talked about like yeast growing on glucose and then producing ethanol, right? The metabolic pathways that cells use to get ATP energy, they're usually shown as like linear, right? And this one's circular. This is what the magic is for these cells. This is why they can grow and make methane at the same time. So here, if they're growing on methanol or methylamines, so CH3X methyl group here, what they do is they take it up, they convert it to methyl coenzyme M. Coenzyme M is this tiny, tiny little cofactor. I'll mention more about that later. And what they do is they oxidize that carbon through these really unique enzymes that were discovered in methanogens. Nothing else has them. It's very similar to acetogenic Clostridia. They oxidize that carbon, stripping off electrons all the way up to CO2. CO2 goes away. Those 6 electrons that are stripped off of a methyl group are then used to reduce 3 more methanol to methane gas. So it goes in this direction. So they add those electrons. And so we call this a disproportionation pathway, because you're taking in 1 substrate here, oxidizing it, 3 more are getting reduced to methane. And the cycle begins again when this terminal electron acceptor is a heterodisulfide of this coenzyme M and then another cofactor called coenzyme B. It gets reduced and then regenerates the coenzyme M that's used to take up some more carbon. Okay, so this is the crazy thing about methanogens. You know how, again, I mentioned like, yeast fermentation and, or if you think about aerobic respiration where we breathe out CO2, that oxygen that we breathe in is accepting those electrons. In this case, in those examples, the terminal electron acceptor for the organisms comes from outside. It's a food product from fermentation, or it's oxygen or something, right? These organisms make their own terminal electron acceptor. So they just keep recycling it. So they only need substrate. They only need this carbon substrate, and that's it, okay? Here I'm showing the example of acetate, and that gets split, and it's very complicated. But there's very few people who understand this pathway really well, so don't feel bad. It's complicated. It's totally unlike everything else. And this is really the secret to then being able to engineer these microbes to do other cool things. Okay, so that's what we call the Wolff cycle of methanogenesis. And for this project that Annalise and Nick were working on, the part that we were trying to speed up is looking at regeneration of this last step here. because you can't do another round of methanogenesis until your electron acceptor is regenerated. So one of the compounds involved in that is methanophenazine. And so that's what's kind of shown here. Methanophenazine is an electron acceptor that goes around and around and is contributing to ATP synthesis by this organism. So if you've heard about quinones before, or coenzyme Q10, we eat it in our diet, vitamin K, very similar to this. Methanogens have a different format, which is a 3-membered ring structure here, and then a long isoprenoid tail. And it acts as this electron shuttle between oxidoreductases in the membrane and the final enzyme that regenerates this heterodisulfide and reduces it to the thiol forms here. So that last step of that methanogenesis pathway. It turns out quinones are found in virtually every organism except for methanogens and then lactic acid bacteria and some of these like solely fermentative bacteria. So those fermenters don't have anything like this. Methanogens have respiration using a quinone, but it's not a quinone, it's this compound, methanophenazine. So we wanted to ask, like, this is a really weird compound. What happens if we mess with this pathway? The first thing is like, well, how much of this methanophenazine compound is made? So we actually figured out a way to extract it from the methanogens. Other people had kind of done this before, but we actually quantified it on a per cell basis, which is super hard to do because you have to be so careful. We want to figure out how many molecules of this methanophenazine are in the membrane of each cell. And so that's what we did. And we also compared it to quinones in E. coli, grown either on LB media, like you usually do, aerobically or anaerobically on fumarate. So E. coli has been known to change the amount of quinones as it's changing from aerobic respiration to fermentation, where it's not doing respiration anymore. And it turns out our methanogens did something kind of similar, which was very exciting to see. Our favorite methanogen, methanosarsina acetiborans, actually produced a ton of methanophenazine. So that was really exciting. It produced just about as much as E. coli grown on LB, even though it's growing anaerobically on methanol. These other methanogens, Methanosarsina barkeri and Methanosarsina mazei, are like cousins of this methanogen, and yet it had very different amounts of methanophenazine in those cells. So that was a really interesting finding from this work. And when we actually plot that out, like how many molecules in like a square micron, or square, I guess, micron of membrane, we can actually figure out like the density of that methanophenazine in the membrane. And it turns out people had figured this out in other enzymes. When you have an electron donor that's next to an electron acceptor, if they're within about 15 angstroms of each other, an angstrom is like 0.01 nanometers, tiny distance, right? If they're within about 15 angstroms of each other, the electrons can tunnel. They don't really hop, but they like ghost jump over to the next one, okay? So people had figured this out in other enzymes. And it turns out that our methanogen, so Methanosarsina acetiborans here, both when it's grown under exponential phase in the culture and in stationary phase, it has so much methanophenazine in its membrane that we can conclude that as soon as an electron hops onto a methanophenazine molecule, it can basically hop everywhere. So it's like quantum tunneling, okay? This happens also in E. coli, grown in stationary phase on LB, but not when they're growing in fumarate. There's not enough there. But we can conclude that Methanosarsina acetiborans can do it all the time, and our other methanogens don't necessarily do it. So what that looks like is, if you think about it, a methanophenazine molecule is floating in the lipid membrane, right? So floating in this two-dimensional surface. And let's say it gets reduced by, it bumps up against an oxidoreductase enzyme, it collects an electron, and then if it's within that 15 angstroms, or I guess it's showing here, if another molecule is within 15 angstrom, it can just tunnel through, just like with no resistance or minimal resistance. And so that basically means this electron, it's kind of like a lightning bolt, can go like anywhere to an electron acceptor or an excidoreductase enzyme somewhere else. So this is what helps to transmit protons across that cell membrane to then make ATP. Whereas if you have a lower concentration of methanophenazine in that membrane, they actually have to do slow diffusion. They kind of have to wiggle again, up to each other to then transfer that electron. So diffusion is super slow. It's stochastic, meaning they have to randomly bump into each other. It has to eventually find its way there. Meanwhile, the energy of that electron is actually decaying over time. Like the longer it's hanging out there, the energy is kind of being dissipated as heat, right? So you're losing energy. You're not producing as much ATP energy if you're losing that as heat energy. So that's kind of what that would look like. But we can say that M-acetoborans has so much methanophenazine that we can know that the whole membrane is basically like conductive, like a metal sheet. It's almost more like a metal ball in that way. An electron anywhere on the surface can get to an electron acceptor somewhere else, anywhere on the surface of the cell. That's super cool, I think.
Is that the first time this has been discovered in bacteria?
So no, yeah. So we know that photosynthetic bacteria have kind of a similar kind of a thing. And we talk about it with geobacter in the subsurface. So there are microbes in soil and sediments that we do know even produce electrically conductive pili, and so have maybe something kind of similar. Yeah. So yeah. So a few examples that was like kind of like hypothesized. So it's not entirely the only one, but it's a big deal to find that methanogens are this way, but it's not all methanogens. And there's reasons if you wanted to read this paper, we kind of discuss. And then so it turns out we were also able to, looking at this pathway, it's not shown here what those genes are for like IP kind of reasons, because it's pretty cool. But we could mess with methanophenazine biosynthesis pathway, and we were able to produce a mutant that had about 20% more methanophenazine, like the density in the membrane was a little bit higher. We had some other mutants that only had minor changes. The blue is wild type, the parent strain. So if you look at the amount of methanophenazine versus the growth rate, this is what that is. So wild type, and then they produce a little bit more methanophenazine and actually grew about 20% faster. And sure enough, if you look at the rate of methane that's produced by these cells, we know that remember I mentioned that 3-to-1 stoichiometry, 3 methane molecules produced per CO2 that's produced, right? They have the same ratio, and these mutant cells grew that little bit faster and also produced more methane. So these are really like super methanogens just by manipulating, I'm going to say, one pathway. And it turns out another thing that we did was then we really kind of tested that prediction. Well, if, endogenous production of methanophenazine affects growth rate and rate of methane production, what happens if we add a mimic, so phenazines, into the culture media? Phenazines are a little bit hydrophobic, so that's kind of tricky, but you imagine maybe if we add this hydrophobic electron carrier, then it might partition to the membrane of the cell, and maybe that could boost them. And it turns out that that's the case. So here we're showing our favorite friend, methanosarcina acetoborans, and we're comparing it to the wild type cells where we didn't do anything. And remember I mentioned how M-acetoborans is already kind of like optimized, right? It's already quantized and it's already conductive. So if you add phenazines, it doesn't help them that much. So statistically, there's like no difference. But methanosarcina barkeri, I remember the one I said, like, it wasn't ever quantized. It wasn't quite as much. For whatever reason, they don't make as much, right? But now when you add phenazine, you get a huge increase. So they're not making enough to make them quantized, but if you give them that little bit of a boost, it actually does improve growth and methane production. So some, we always think about skeptical reviewer number 2, who's going to like hate on everything that you do. So one thing we were thinking about is like, a skeptical reviewer might be like, oh, how do you really know it's getting into the membrane? You're just adding to these compounds. So this compound, 5,10-dioxyphenazine, super chemically similar to this compound. And in this case, but what happens is because it has these 2 hydroxyl groups, if it gets into the membrane and accepts an electron, it creates a really toxic kind of like radical. So these dioxy groups kind of stabilize it. And then when you have a radical in the membrane like that, nasty chemistry can happen. Like you can cross-link your lipids. It's like not happy, right? And it turns out we can show that indeed this toxic compound must be getting into the membrane and must be accepting electrons, because we do see that it does become really like toxic. So at a very like low concentration, and it's more toxic for methanocyrsina than it is for M-acetoborans. So we can conclude that if you exogenously add phenazines, they must be getting into the membrane. Once they're in the membrane, they can accept electrons. So that's what we showed that. Okay. So another cool example of how we're using methanocyrsina. So we do a lot of basic science like what I just showed you, but then we also start to think about how can we extend that into translational or biotech kind of uses. So this project was carried out by 2 grad students in the lab, Jared Aldridge and Sean Carr. And Sean is actually now a CEO of a startup company here in Lincoln called Molecular Trait Evolution. So when we think about methanogenesis, this crazy pathway, right? One thing you can notice, I mentioned it before, that you have these like super cheap, low energy substrates, hydrogen, acetate, methanol, methylamines. These are the cheapest substrates. And if you're going to have a biotech use, you want your feedstock to be cheap, cheap, cheap. Okay. You want it to be waste. You want it to be trash. Okay. And so these have such low energy that they're very inexpensive feedstock. And this is where methanogens really kind of shine, because they're the champs at consuming these low value substrates and then of course making methane. But what if we could get them to make something besides methane? So we were kind of thinking about what other bioproducts could we get our methanoarchaea to actually synthesize? As if we could do that, we already know that we can use methanoarchaea in wastewater treatment plants to make biogas or compressed natural gas for renewable energy sources. So you can imagine like, hey, we could just use that same thing, except this time get out another high value co-product. So we were like, what could methanosarsina make? Well, like where would it have kind of an advantage? And we were kind of thinking that methanogens would be really good at making isoprenoid lipids or terpenes. And this is because what you learn in your textbooks is that bacteria and eukaryotes, right, have membrane lipids that are primarily, so this is an example of like how much of an E. coli cell is protein versus carbohydrates. And then their membrane lipids are all fatty acid membrane lipids, right? And there's only a tiny amount that are producing isoprenoid lipids. It turns out, though, that this is a defining characteristic of archaea, that archaea, this is showing for Methanosarsina barkeri, produces a lot of protein, kind of proportionally less carbohydrates. They really don't produce any fatty acids at all. Their membranes are all these isoprenoid lipids. And so that's kind of shown here on the right. So Methanosarsina produces actually very, not very complicated lipids. If you have looked at like what types of fatty acids plants will make, like for canola oil or something like that, there's like all these different saturations and hydroxylations and things like that. Methanosarsina acetoborans actually makes relatively uniform isoprenoid lipids. So it's kind of like a blank slate if you're thinking about engineering new types of lipids. And it's also really awesome because these isoprenoid lipids are very interesting for producing things like squalene, which might be like your very nice face creams, or things like they can be used for like adhesives and for what do I want to say? Lubricants and stuff like that. And these isoprenoid lipids are actually much better than the fatty acid kind of lipids, which might be great for our diet, but functionally are not always ideal. So we were thinking this is a good candidate. And so we thought, well, maybe we can get our methanogen to make isoprene, which is a hemiterpene monomer form of that eventually turns into those longer chain lipids. But what about if we could like overexpress or overproduce that monomer and then recapture it? So this is an example of a few years ago. We just did this. Most isoprene industrially used, of course, there's natural rubber. That's what natural rubber is made out of. And then synthetic rubber is from petroleum. It's the light crude fraction. And so that's used to make rubbers, polymers, adhesives, different types of tires, or butyl rubber, or things for like medical uses. And this hemiterpene monomer is part of epoxy hardeners, flavorings, and other types of drugs and all types of things. And that's about a $4.3 billion market per year worldwide. But this actually doesn't include gasoline. The primary component of gasoline is isopentanol, which is just a like hydroxylated form of isoprene. So like you can't even find how much isoprene is like gasoline is worth, right? It's just huge. So any kind of production of isoprene by our methanoarchaea would be potentially a pretty big deal. So we decided to go that route. So using our molecular biology and biochemistry knowledge, we created a plasmid that produced the plant isoprene synthase gene, on that plasmid, and we integrated it into our methanogen's genome. And when we first looked at growth on methanol, we were kind of sad because we didn't see any difference. If you're thinking like the membrane are made of isoprenoid monomers, and you're going to take from that pool and produce this isoprene compound, which I forgot to mention is highly volatile. It's like a gas. So like that would go away and go bye-bye, right? So that would be a problem for the cell, right? They're not going to make any membranes. So we expected them to be kind of limping and kind of sick, but we didn't see that. But, we're good scientists, so we're like, well, why did it break? How did it break? Maybe we can figure it out so we can fix it. So we did an enzyme assay, and we could see, though, that our gene did look like it was active. So we saw this pyrophosphatase activity indicating that there was more being synthesized, there's more activity in the cells, and it did correlate with our integration of our plasmid onto the genome. So it's like, well, it seems like we added it, and it seems like it's active if you're assaying for it directly. So then we looked at, well, maybe we should just see, is there isoprene actually being produced? And it turns out there's a lot. So we were able to detect in our bulge tubes about 1 millimole of isoprene that we could detect. And this was through gas chromatography with an authentic standard. So like, that's pretty good evidence. But we were so like shocked that we actually then also did mass spec with it. So, and indeed, it's authentic, matches the mass exactly of isoprene. So how come it's growing okay? Like that was like, how could it possibly be making quite a bit of isoprene, but actually be growing okay? Was not what we expected at all. So then we did Jared and Sean did really, really hard experiments, what we call mass balance experiments. So because our methanoarchaea grow in sealed anaerobic tubes, we can add a precise amount of substrate and then measure all of the products. So we can actually do the math to figure out where did that carbon go. So what we saw is that all of the methanol, as we would predict, was consumed. But our isoprene synthase expressing cells seem to produce less carbon dioxide. This is also not what we would have expected. And we saw maybe a little bit of a decrease in the methane produced, but this is not a statistically significant amount that we could determine. So it seems like the isoprene that we were detecting all came from this CO2 pool. So they didn't, remember I said in the pathway that substrate is getting oxidized to CO2. So they're not making it all the way to CO2. They're coming off as isoprene before that step. So when we plot that out, all that mass balance data, this is what that looks like. So 100% consumption of the methanol, 96% but not significant amount of the methane being produced. This is telling us like the same amount of ATP is being made because all the electrons are getting to where they need to go. But instead of oxidizing all the way to CO2, you have 4% of the total carbon that's going to this new isoprene co-product. So there's still methanogens, but now we would maybe also call them methanoisoprenogens because they're now producing two products, but this is all consistent with them making ATP. So 4% flux to isoprene sounds like maybe that's not, I mean, I was pretty excited about it because that's on a per carbon basis. That maybe doesn't sound like a ton because, 1 millimole doesn't sound like a lot, but if you think about it, other people who have engineered yeast or E. coli to make isoprene like this, where they've engineered it, they had to put whole new pathways in there, tons of genetic manipulation. And what they don't say is they actually have to force feed the cells like so much glucose to get out the high yields of isoprene. In this case, we added one gene and it's already at 4%, and that's on a per carbon basis. So it turns out this is actually more efficient than the other engineered organisms that people have worked with. And so this is also a super big deal that our cells are actually, they had a tiny bit higher biomass that we could detect. So they're actually better by making this isoprene. And that was also kind of unexpected, but in retrospect, we can rationalize it because it's kind of hard to grow as a methanogen. So if they can like spit out this other volatile product and like get it out of there, and they're still able to recycle their electrons a lot faster, they're like, great, happy to do it. So they're actually able to grow faster and a little bit better by actually co-synthesizing this isoprene. So now we call them methanoisoprenogens. And then one of the things that people, okay, so when we were working on this project, people were like, oh, but that doesn't matter because there's no way you're going to be able to do it at scale. People will use methanogens to treat wastewater. So they'll collect up all that municipal waste or whatever, or manure, right? If you hear about biogas production and anaerobic digestion, that's all methanogens in there, the wild methanogens. So people were like, you're never going to get your engineered methanogens to compete in that crazy mixture of everything wild that's in there and fungi and God knows what, right? So we're like, okay, well, let's do the experiment. So we actually inoculated our engineered methanogens into wastewater anaerobic digestate. And what we could see is that, yes, if we add methanol substrate, they still produced methanol. We didn't see any change with methane production. So everything in there is happy. They're still making methane. And if we don't add, and we also designed a synthetic wastewater to kind of sort of mimic this, but have it be like sterile. And so if we don't add substrate, you don't see anything being produced. But then when we look at isoprene production, only when we add our engineered strain, we were actually still able to detect isoprene in the headspace of these little microcosms on the wastewater digestate. And for these two, we actually added methanol and because we're sort of like, priming that, giving them the substrate that they love. But even when we didn't add any substrate at all, we were still able to detect a little bit of isoprene in the headspace. So these engineered methanoarchaea are actually capable of competing with those wild methanogens and whatever else there, and they're still able to synthesize that isoprene. This is super cool because we're showing that we can use our engineered methanoarchaea as like potentially in future as a drop-in to anaerobic digesters, and you'd be able to get out this isoprene. So this opens the door to not just isoprene, but whole families of high-value compounds that are made from that same pathway. Another group in Germany has shown bisabolene can also be produced in good numbers from our methanoarchaea. And so we're pursuing all of these other options. So these are simple related terpenes. Pinene is like the pine sol kind of smell. Geraniol is kind of a rose smell. Zingiberine is like the ginger smell. So these are highly valued as components for drugs and also for flavors and fragrances. And then in the last couple of minutes, I wanted to tell you about how we can then take knowledge from these methanoarchaea and think about how to apply those tricks to other organisms to improve them, right? So again, microbial physiologist, I'm obsessed with metabolic efficiency and how do I like tweak things, right? So methanoarchaea are super, super efficient at managing electron flow and making that ATP, right? So like, is there something that the methanogens do that we can apply to maybe plants or something and get them to be improved? So this is work done by grad student Connor Hines, and then 2 undergrads in the lab, Alicia Salvi and Brandy Russell. Alicia actually works at Zoetis now. So one thing we thought about going back to this coenzyme M molecule. So coenzyme M is this tiny cofactor all the way down here. So this is the one that does that C1 carrying, that carbon carrying in the methanogenesis pathway. And there was biosynthesis pathways in some of the methanogens. These enzymes are highly anaerobic or so sensitive to oxygen. And then in our methanosarcinales, it seemed like they had a slightly different pathway. And then the last step to go from this compound to that coenzyme M was really not very well known because it also can happen autocatalytically. So there's still some questions about the biosynthetic pathways in these methanogens. And so we wanted to see, like, you know, this tiny compound has a thiol group, and that automatically makes it an antioxidant. So we were kind of wondering if this tiny antioxidant could be added to other microbes or plants to then actually protect them from oxidative stress to get them to be more metabolically efficient. So coenzyme M, by the way, is also already a World Health Organization essential medicine. It's actually given to people undergoing chemotherapy to protect their kidneys from aldehyde toxicity. So it allows you to take up more of your chemotherapy drugs to kill the cancers, but then doesn't kill your kidneys. So it's actually a pretty safe compound. It's been highly tested in humans and animals. So we wanted to see if we could synthesize it endogenously in E. coli, and we were actually able to do that. So we designed a synthetic 2-gene operon, which we called ComSyn. So using our crazy biochemistry knowledge and non-model organisms, figured out this new pathway that doesn't exist anywhere else in nature. And we're able to show in E. coli lysates that we could synthesize coenzyme M. And it turns out that when we put that operon into E. coli, it actually does increase resistance to oxidative stress. It likely supplements the endogenous glutathione system. And we designed it to be dependent upon addition of taurine as a co-substrate. So this is kind of how that pathway works. You add taurine, and then it uses these genes to make coenzyme M. And then when you have that coenzyme M, it can protect from just cellular byproducts of oxidative phosphorylation, like hydrogen peroxide and hydroxyl radicals that are produced in the respiratory chain. And then it turns out, and we've known this, that coenzyme M, when it's oxidized, can form these disulfides, or it could also form a disulfide with the glutathione system. And then because it was already characterized in humans and animals, we know that our glutathione regenerating system works on coenzyme M too. So we already knew that. So it worked really well in E. coli. So then we were thinking, well, what other organisms? And of course, being in Nebraska and caring about plant biochemistry in our department, we know that one of the main limiting things for plant metabolic efficiency is reactive oxygen species, right? They produce so much in their chloroplasts, and they spend a lot of energy repairing that stress. So we're kind of wondering if we could actually apply our coenzyme M. Just, it's hard to engineer plants to synthesize new genes and stuff, right? So we're just like, what if we just spray the plants? Okay, if there's any hope that it will work, if you spray the plants with it, we should see something, right? So we actually then sprayed our coenzyme M onto these plants, and then lo and behold, they indeed grow quite a bit faster. So this is a dose response curve. You see no treatment, so the rosette area of those Arabidopsis plants, and just with 0.2 millimolar, you start to see huge improvements in growth rate of these Arabidopsis seedlings. So not only do the leaves grow bigger and out faster, but actually the roots actually grow longer, faster. That's very exciting. We see that the chloroplast is electrically reduced, which is what we would kind of expect because they're protecting from that oxidative stress. And it does seem like our coem, if you put it in the culture media or if you spray them, it's actually also even better than if you sprayed with additional glutathione. So glutathione is plants, just about every organism, not methanogens. Every other organism produces a lot of glutathione. So even if you boost it, it's still coenzyme M is actually better on a per molecule basis. So they have higher wet mass and significantly higher dry mass. So we're really excited about this. We file patents on it, both on spray applying it, but also engineering E. coli to make it. And this opens up this is the proof of concept that if we could, next step would be putting it into the chloroplasts of plants, because if we can produce it endogenously where that's happening or in other tissues, it also could potentially be helping with iron uptake from the soil, just because of its chemical properties. So if we could target it to express in different tissues of the plant, we would expect to see improvement in growth and metabolic efficiency of plants as well. And I didn't have a slide in here, but we also did test this. It actually works not just in Arabidopsis. People don't get that excited. But it works on basil, cannabis, and also we have some good data. Soybeans are a little bit funky. Not all of the trials worked with soybeans, but we did have one greenhouse trial where the ones that were sprayed have 50% more soybeans. What? Yes. Cray. So we're trying to figure out, like, why did it work so good that time? We do think that it's really, really helping when these plants are under lower light conditions. We seem to have, like, if things were planted more towards the fall in our greenhouse, it was better. And that's exciting because a lot of, crops, the high value cash crops are actually grown indoors. So, or, tying it to space travel, if we're going to have a thing on the moon or go all the way to Mars, you're going to have to grow under low light conditions and have it be efficient and nutritious, right? So pretty exciting. So we think we have some really good insight into how to produce, improve metabolic efficiency of plants. And our colleague Kasia Glowacka in our department, and Alini and Jeremy, so we have hopefully a publication coming out soon, but they were actually able to show that it does affect non-photochemical quenching, which is telling you that the cells really are being protected from oxidative stress. Oh, so sorry, this is that data. Yeah, so it works on cannabis, basil, and those were one of our soybean trials there. So in all of these different plants, it's actually improving metabolic efficiency, and that's really exciting.
And you sprayed your coem from the bacteria, or this was just like a pilot trial with like coem that you purchased?
Yeah, so this is a pilot trial. What I would be really excited about is we want to maybe see if we can engineer rhizobacteria to produce coenzyme M to help protect the rhizobacteria from oxidative stress to then make more nitrogen fixation for the plant, as well as engineering the plant to produce it. Yeah. So and there's a lot more to do, like, so the E. coli synthesis, so we can detect it. being synthesized in an assay, but it actually is really, really tricky to quantify how much coenzyme M is being made in E. coli cell. Thiols are really, really hard to quantify and deal with because they oxidize like crazy and stuff like that. So there's potentially a lot more that we could do to optimize to get more production. But yeah, that would be another situation of like, it'd be great if we could like really produce a ton of it naturally with the E. coli and then spray that on as like another fertilizer or something. Yep. Lots of cool things that we could do. So those are the just thinking about the future directions. So we mentioned that coem, which is a methanogen trait that we're improving other organisms with. But it's important to remember that even just with methane, these methanoarchaea are producing methane gas at a 60 to a 90% conversion efficiency. And so all around, right, like we see the big ethanol fermenters, those yeast are only 12% efficient. Methanoarchaea are like 99% efficient. So they're incredible from a biochemical perspective. And if we can control or scale up even further, we know that we could put that renewable natural gas, that renewable methane into our natural gas infrastructure. So we already, produce compressed natural gas. There's nothing that says that you couldn't do that for renewable natural gas from an anaerobic digester. So that's what we're talking about. And methane is actually a rocket fuel. So these microbes grow by producing this rocket fuel. So you can imagine not just compressed natural gas shown here for things like trucks and cars, but also for launches into low Earth orbit. And so there's plans, Department of Energy and our country's leadership have already kind of counted out that the US alone produces 364 million tons of biomass. And what we're really talking about is using engineered microbes to valorize that, sure, to make a lot of other chemicals that we would need. But to do all of that, you need energy. So it's almost like regardless of what microbes you're using, we're going to have to use methanogens to produce renewable natural gas and potentially other high-value co-products to power that fermentation and also to clean water and to produce fertilizer.
We need like a reactor of methanogens to fuel the other reactors.
Absolutely. I mean, otherwise you're competing with food, right? If you're eating those, if you're using starches and glucose, it is going to get to a point where it's competing with food. So what we really want to do is focus more on valorizing the waste, things that we can't eat or that our livestock can't eat. How can we turn that into high value products? Yeah. Great. So, and with that, I'd like to mention my students. So these are my current students and postdoc, and then all the graduated students. And I mentioned everybody in bold contributed to the stories I told you about. And these are our funding sources. So lots of funding throughout the years, primarily through NSF, also USDA, Department of Energy, some NIH funding, and then generously supported by Nebraska Center for Energy Sciences Research, NSF EPSCoR, the Corn Board, the Soybean Board, and the Water, Environment, and Reuse Foundation. And the Energy Center is funded by Nebraska Public Power District. And then I have two startups. So one is for consulting, and then this is Molecular Trait Evolution, which is headed by Sean Carr. So he took a totally different project and is scaling that up as we speak. So with that, I'd be happy to take any questions.
Have a question?
About molecular trait evolution? Sure. So it actually was technology that UNL had for over like maybe 20 years. So we formed this startup with Paul Blum, who's now an emeritus professor at UNL. And for that project, we actually found or produced an enzyme cocktail that can dissolve corn starch, corn fiber, so lignocellulosic biomass, also So that's like all of the, you know, all of the seed materials can dissolve them. Uh, and it also works on soybeans and on nut shells. And so it releases, the cellulose fibers are sugars they're connected into fibers, right? And so these cocktail enzymes can break that up into smaller pieces to make a lot more sugars available for the yeast that might do fermentation later on. But also because it's dissolving all of that, turns out it helps to make it easier to access the oil from like your seed crops and stuff like that too. So processing enzymes, so it's a proprietary cocktail, and it turns out it works better than what's commercially on the market right now. One of our, because I mentioned we had this cocktail for a long time, we actually had enzymes at 4 degrees for 5 years and they were still active. So super stable enzymes, and it turns out that you can even boil them and they retain activity. So that's really exciting for ethanol and bioprocess condition, because the major cost for ethanol production is the processing enzymes. So if we can recycle those enzymes or recycle the liquor, the water, we're hoping we can drive down the water use for ethanol production. We're hoping that we can help fermentation from other waste feedstocks as well, not only corn, although corn is great, but lots of other things. And make it more efficient also to extract biodiesel, the oils, right? So cross your fingers. So MTE is doing pretty great and lots of innovation, more innovation that we want to do. But it'd be really exciting to have a Nebraska source of these enzymes that we could use really broadly for biomass processing.
Is that cocktail just cellulases or does that include?
It's proprietary.
Just trying to stay general.
Yeah. And then like I'm the researcher side, so I'm like, there's so many more other cool things we could do to make it even better, right? But we have it to the point where we're moving it as a product now since it already works better than what's on the market. But yeah, if we can continue to get funding to continue to innovate, that would be really great. Yeah.
Anybody have any questions?
I always ask this question, Nicole, because you've seen a lot of students go through your lab. What sort of habits or characteristics do you find that make students successful in the STEM field?
Persistence.
Okay.
Being really curious is really important also, because sometimes you can have the tendency, okay, this is my project. I'm just going to like work on my project. But to really be an innovator, you need to know kind of what everybody's doing and to have like all of these ideas, by the way. So the COM project, when we presented that at a national meeting, I had people coming up to me being like, how the hell did you think of that? Because they've known about this compound. Nobody had ever applied it to plants. They did millions of dollars of work to show like the pharmacology, how it works in a human and stuff. Nobody ever tried with plants. So just, you never know where that new insight or inspiration is going to come from. And or if you have a roadblock, it's still biology. It's still complicated. There's still a lot that we don't know. So sometimes the way, over or through a problem can be unexpected, where you're just like, oh, that chemical is kind of similar to that one. Maybe this will work. Or maybe you want to, engineer a new enzyme or look in a new place that you never would have thought. So actually, I do feel that like AI is maybe not as much of a threat as we think it would, because frankly, like a lot of this stuff was me at 2 a.m. being like, oh, what about that crazy thing? How did I know about that? Because I read a paper 20 years ago and it just popped into my brain, literally. Okay. So if as a student, you want to read broadly, you want to go to those seminars, because like 5 years later, 10 years later, you have no idea where you'll make just not even a good connection. It's not like going to be, oh, it's the same compound, right? It's going to be like, it's kind of similar, sort of. And then maybe you can find a connection, right? So but being persistent. You have to not give up. You have to be a really hard worker, persistent, hard worker, curious. And there is a lot of also being super honest and trusting your PI. A lot of these projects were side projects where it's like, what about this crazy thing? Let's just like try it and then see. And then it turns out like, oh my gosh, it's like way more interesting than we even thought. Like it was going to be kind of cool and it's like super cool. So, uh, all of those things, kind of together. It's really good to have a support system. Science is really hard. We fail a lot, and there's a lot of like criticism of like, oh, well, you didn't do that, or oh, but it's too expensive. That feedback is actually good because it helps you to improve things, right? Unless somebody says, like with our wastewater anaerobic digester part of it, right? That was the criticism. It's like, oh, but you'll never be able to get to do that. Well, let's do the experiment, and that will quiet them or not, or we'll know, right? So not being afraid to take that test or kind of to take on your critics a little bit. If it didn't work out, then we'd know. Maybe we would switch to a different organism. Maybe we'd think about something else about how you design a bioreactor or something. But unless we did that experiment, we would never know, right? So you can't move science forward unless you're willing to tackle those difficult questions and not to take it personally because it's science. It's not, you're not a good or a bad person. It just is, right? So we do the experiment and find out what it is, and then we will move from there. So, yeah. Yeah.
You see this a lot, you see this being a lot more like relevant or important because of like the global like supply chain issues with crude oil.
So, I mean, that's maybe top of mind right now. But energy has always been important throughout human history. And I happen to know about these methanoarchaea because I happened to do my PhD in a lab. My advisor had worked with them and was on some of these very early papers in the 80s. So there's like random kind of introduction to it. But I actually, when I sat down and thought about like where I wanted my career to go, I could have gone into biomedical, but the way I kind of see it, and that's really important and that's great. But unless we have people with these sets of skills, we can't ever get to like renewable energy, right? There are issues with wind and all those other sorts of things, but we already know that these are super efficient. We already have them scaled up worldwide. So every mid-sized city has a wastewater treatment plant that does this, and that's with no optimization. That's just literally, a tank, right? So there's tons of potential there. So no, I mean, there's a lot of interest because of what's going on in the world. There's a lot of interest because of global climate change, but you might not realize it, but these organisms are in the subsurface. So when I started my job, it was thought that like, oh, that natural gas was all fossil and that was like abiotically produced. But that entire time I was like, I don't know, because they're in the subsurface too. Like, I bet they're actually making what we think of as fossil. It's old carbon, but there are methanogens down there. And in the intervening 15 years, it turns out people are like, oh, maybe a huge portion of it is actually biologically produced, maybe even most of it. It's just been trapped and we haven't gotten there, right? So if you think about it, methanogens are already active around the world in the subsurface at scale. These are the things that are producing most of the natural gas. So what we want to do is accelerate it with younger carbon, up here, not all the way down there. So in that sense, they've already been important. The reason we don't burn coal in the United States is because they figured out how to frack natural gas. So it is because of these organisms that we don't have acid rain and lung problems. So it's much cleaner burning. So as a researcher, though, as a professor in particular, we actually don't live in the present day. We live like 20 years from now. Like everything we do is like, what do we need 20 years from now? We got to get started today. We got to train you people now so that 20 years from now, when you're, middle-aged and are making decisions, that you're all like, have the knowledge and the skills to tackle new problems. So we really live kind of like in the future. So this is where back when I was a graduate student, that's kind of where my brain went. I'm like, energy is always going to be important. Renewable, sooner the better. If we can control that, it's going to be beneficial to everyone. So, yeah.
Have you tried like spraying genetically modified plants? I know you mentioned that you were thinking about maybe some modification to the chloroplast.
Yeah, so that's tricky. People get really nervous if you're like, I'm going to spray microbes on your plants and stuff, right? So they actually, from what I understand, they do that commercially, but it's only for like highly regulated like strains of microbes and stuff, or they might not be genetically modified. So there are rules about that right now. But we have learned a lot about genetically modified organisms, you know, in the past 20 years, right? There are only, I think, 4 genetically modified organisms that are approved for like use or release, but we've been studying them for 50 years, right? So right now, the government is actually trying to think about, should we revise our laws? Should we expand the number of organisms that are regarded as safe because we know they don't make anything dangerous? Should we, or can we think about a better way to assess, like if you're going to do synthetic biology, move genes across? Maybe we don't care as long as it's not a pathogen gene or not like immunogenic, right? Which is most things. Most things are not going to be immunogenic. They're not going to be pathogenic, right? But how should we assess that? Maybe we want to have a system for evaluating, like if I did spray this GMO microbe on these plants, can we do an assay to be like, hey, if we did that, we know that these microbes are going to die after a season or a few months. Like maybe they'll have the effect, but then die, and then so we don't care, right? Versus you probably don't want to engineer something that's going to persist in the environment for forever, right? So right now the government is trying to think about maybe we should change the way we regulate things. Other countries actually are not as strict, by the way. So, and they just did this like big assessment. So in other countries, there maybe isn't something stopping someone from making a GMO and spraying it on whatever. In the US, we regulate it quite a lot. So, but ultimately in the future, yeah, we would want to test that. Everything that we do now is in the lab and we kill everything when we're done. So it's just that point of research, like, is it possible? Can we do it? If we could, what would that be like? But we're not going to break any laws, right? We're going to follow the rules always. Scientists follow the rules. We like to make rules and then we follow them. So.
Awesome. Well, thank you.
Okay. Thank you so much.
For those that joined us online.
And
Oh, yeah. Yeah, if you would like to take a nice survey about, give us some feedback about either this seminar or if you've attended other seminars, we'd appreciate that. Always try to come up with new ideas or make things better. So scan this.
for people that being part of us, this has been an effort to show what is going on in Nebraska. Our goal is to do it better and to do it the best and to spread all these amazing people that have passed over here with all this new knowledge. As Dr. Nicole said, we don't know what's gonna come out after.
Yeah, somebody's gonna have it in 5 years from now. Someone's gonna have a genius idea because of this. That's what's gonna happen. Yeah.
And the survey is because this is grant funded too, so we need to and to present what we doing.
Yeah.
And thank you for your company.
Yeah. And take cookies if you want to for the road.